Hello everyone, this is Akshay over here and welcome to Akshay's Biology classes. So as you people might have noticed that I have not uploaded any new videos recently. Regarding that, I am extremely sorry because I was quite busy with some of my personal work. But however, I will make sure that from now on, I will try to be more consistent and regularly upload new videos and new contents. So with that being said, I have actually planned few videos on immunology, few series of videos on immunology where we will try to understand some of the core concepts and core ideas of this particular topic, that is immunology. And with that being said, the first video of this particular series is on hematopoiesis. So without any further delay, let's get started. Now, hematopoiesis, as many of you people might have heard this in the immunology, and in some cases it's also studied in the developmental biology also. So now, to define hematopoiesis, it is the generation of blood cellular components. Now, what do we mean by the blood cellular components? It is basically the red blood corpuscles, white blood corpuscles, and also the platelets that make up the blood cellular components. So these three cell types is called as the blood cellular component. So the generation of these all three blood cellular components is called as hematopoiesis. Now all of these three different type of cells in the blood are actually derived from a single cell, that is the almighty hematopoietic stem cells. So hematopoietic stem cells are pluripotent stem cells, the ability to produce or give rise to all the three germ layers and also multiple lineages of cell types. Apart from this, there is quite an interesting property of the hematopoietic stem cells, the self-renewal property. It means that in the body, the hematopoietic stem cell population never goes out dry. It is never completely exhausted. In other terms, in other words, a constant hematopoietic stem cell population is always maintained. That is, the hematopoietic stem cells undergo mitotic division continuously to produce new hematopoietic stem cells. Now this makes actually sense because all the white blood cells, platelets and RBCs are produced throughout one individual's lifetime. So to produce new RBC, WBC or be it platelets, obviously there should be a stock hematopoietic stem cell population. And to have a stock hematopoietic stem cell population, the population should be always maintained, and this maintenance is actually done by the self-renewal property. So now let's move on to the site of hematopoiesis, in the sense where does the hematopoiesis actually occurs. Obviously the answer is bone marrow. But however, during the developmental stages or during the fetal stage, we see that liver is the primary organ for the hematopoiesis. If we try to question where does the hematopoietic stem cell population in the liver arise from, so the earliest site is the blood islands of the yolk sac. It is the site where the first few hematopoietic stem cells actually form, and the hematopoietic stem cells which have actually formed in these yolk sac, they migrate to the liver, and at the time of birth, when the bone marrow is completely constructed, when it is completely ready to take over the hematopoiesis, the hematopoietic stem cells from liver, they migrate to the bone marrow. But however, there is a considerable controversy whether these hematopoietic stem cells that come from the yolk sac actually populate the fetal liver. Some of the advanced studies, mainly through the transplantation studies, it has been revealed that two stem cell population actually exists, and one of the population is derived from the yolk sac, and these stem cells basically form or produce the blood in the case of embryo, while another stem cell population is actually derived from the lateral plates planchinoplurae, which is also referred as the aorta-gonadal mesonephric region. And it is quite interesting to notice that some of the endothelial cells in the sense we are mainly speaking of the blood vessel cells, right? The endothelial cells of the dorsal aorta also produces some unique cell population, and this unique cell population gives rise to the hematopoietic stem cell. And with the advanced invasive techniques into the embryo, we have actually identified this unique cell population. This unique cell is actually termed as the hemogenic endothelial cells, which gives rise to the hematopoietic stem cells. And these hematopoietic stem cells migrate to the liver. Now, there is a transcription factor termed as RUNX1, and the main function of this transcription factor is controlling the transition from the hemogenic endothelial cell to the hematopoietic stem cells. So if you inhibit this particular transcription factor, we see that no blood stem cells are actually formed in the yolk sac, umbilical arteries, dorsal aorta, and even the placental vessels. So what is the takeaway? What can we conclude from all these studies? It is said that during the developmental stages, liver is the primary organ for the hematopoiesis. But the stem cell population that is actually existing in the fetal liver is coming from the placenta to some extent, the AGM region, that is aorta-gonadal mesonephric region, and also blood islands of the yolk sac. But however, we are not yet sure whether the yolk sac is contributing for the hematopoietic stem cells. So this is the ultimate takeaway that we can give from this particular part. So now let's look on to the process of the hematopoiesis. As we all know, the process actually starts with the almighty cell, that is the hematopoietic stem cell. And hematopoietic stem cells will give rise to two progenitor cell lineages. The first is called as the common lymphoid progenitor cell, and one more is termed as the common myeloid progenitor cells. So the common lymphoid progenitor lineage gives us three different cells, that is T cell, B cell, and the natural killer cell, or also called as the NK cell. On the other hand, the myeloid progenitor lineage will give us granulocyte macrophage progenitor cells, and another lineage we're getting is the megakaryocyte erythrocyte progenitor cells. The granulocyte macrophage progenitor cells will give us the neutrophil, eosinophil, basophil, mast cells, and the monocyte. Remember, the monocyte is present in blood, but during a local injury, the monocyte enters into the tissues, and after entering into tissues, monocyte differentiates into macrophage. While the megakaryocyte erythrocyte progenitor cell will give us the megakaryocyte and the erythroblast. The megakaryocyte will give us the platelets finally, and erythroblast gives us the erythrocyte, or in other words, it is the RBC. Now there is one more cell called as the dendritic cell. Dendritic cell is also called as the antigen presenting cells. Now what is an antigen presenting cell? We will study this again in the upcoming videos. The point here is that dendritic cell is quite a unique cell because dendritic cell can be derived from either common lymphoid progenitor cell or even from the common myeloid progenitor cell. In other words, this dendritic cell can be derived from both. It can come from either myeloid progenitor or can be from lymphoid progenitor cells. So this is the flow chart that tells us about the hematopoiesis. So let's stop our session over here, and in the next video we will try to understand the hematopoietic inducing microenvironments and transcription factors, and finally we will also look into the regulation of hematopoiesis. So we will meet in the next video. Till then, goodbye.